Hey, it's Mr. Peterson. This screencast is about DNA replication. So DNA makes an exact copy of itself when cells divide. And so we're thinking about mitosis, cells making more cells, either for repair or growth or whatever reason that cells are dividing. So DNA acts as a template, it's a pattern. So the same rules, the A's and the T's and the G's and the C's, have to match up. It happens in the S of cell cycle, and the DNA copies are identical. So here's that business of the cell cycle and the business of mitosis. So mitosis is M. So at the end of mitosis, after cytokinesis, cells jump into that G1. Then they get the nod. They get the instructions. It's time to divide, and the cell will then undergo DNA replication in the S. So DNA synthesis of the cell cycle. G2, a little bit more growth, and then finally jumps into mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis. And each one of those cells that's produced by cell division will have an identical copy, or it should be an identical copy of DNA. So those are the things that need to happen for replication to occur. So here's a strand of DNA, and we're going to look at animation that's going to simplify the process. So here's our double helix. We're going to zoom in. The molecule gets pulled apart, gets zipped apart by some proteins, by some enzymes, and those are the models, and those are simplified nucleotides. So then a protein comes along, DNA polymerase, and it builds new strands of DNA. So now we've got two complete strands of DNA. We have an original strand and a new strand. It's called semi-conservative replication. So if you recall, that would be in S, and S makes chromatids. So now that's how chromatids are made. So DNA replication. So it's a simplified model here of a really, really complex process. And we're going to kind of keep it about simple. So DNA replication requires a pattern. And that's one of the unique things. If you remember, one side of the DNA is a mirror of the other. So you have A on one side and T on the other, and G on one side and C on the other. And the work is done by enzymes and some other proteins. And the first thing that has to happen is that enzymes called helicases, hear that helix, helicases split. They pull apart the DNA. They break the hydrogen bonds between A and T and G and C. And then free-floating nucleotides are plopped into place by another molecule, a molecule called DNA polymerase that does the work. And so enzymes and proteins, if I use those things interchangeably, it's because they are. Enzymes are proteins and proteins are enzymes, and those are the things that do work in cells. So the enzyme we're really concerned about with here is a molecule called DNA polymerase. Now, we use DNA polymerase in our cells, and so do bacteria and fungi and everything else. Because remember, DNA structure is the same in all organisms, so it requires the same molecules, the same kind of molecules to do the same kind of work, perform the same functions. And those polymerase enzymes have the ability to build covalent bonds between the nucleotides, between the sugar of one nucleotide and the phosphate of an adjacent nucleotide. It requires some energy, requires some chemical energy, and so here's a snapshot of the DNA polymerase taking a nucleotide and adding it to the template, so it matches up and it builds a new strand. And so those complementary bases pair up and they form a new strand. So those proteins and enzymes perform something called semi-conservative replication. So if you remember in class, I gave you those nucleotides, and some of you took your DNA and you split it this way, and you tried to make copies that way. When we know that the DNA actually unzips and we wind up with a semi-conservative model, what does that mean? That means half of every DNA strand that gets replicated is new, and half of it is the old template that was the model for the production of DNA. Now, the DNA is really long, right? In every cell, we get like 6 feet of DNA. And so in eukaryotic, so cells like ours that have a nucleus, eukaryotic DNA replication, we have a bunch of different places that DNA will get copied all at the same time. So that if we start here and we go down, as you go down from A to D, those origins of replication, those little bubbles, are where DNA is getting copied. In eukaryotic, so cells like us, our DNA polymerase molecules can throw down about 50 nucleotides a second. So 50 nucleotides a second. And in every cell, if we're making a copy, there's 3.2 billion nucleotide pairs. It's going to take a while for DNA replication to happen in humans. If you're a bacteria and you only have about 8,000 base pairs, or maybe you've got 50,000 base pairs, and you can do it at 1,000 times a second, it's not going to take very long for you to make copies of your DNA. And that's one of the things that happens with eukaryotes and prokaryotes. It's a little more complex because we've got chromosomes. Our DNA is in chromosomes, remember. And one of the cool things about DNA is that if mistakes are made, DNA polymerase will proofread. It will make corrections. And so DNA replication happens so that cells can divide. It happens pretty quickly, even in eukaryotic cells. 50 nucleotides per second is pretty quick. And if we've got multiple points of replication across chromosomes, it's going to be done in a matter of hours, but still not as fast as bacteria at 1,000 nucleotides a second. So DNA replication, really important event in multicellular growth of multiple copies of cells. DNA replication. Hope that helps.